Hello everyone, my name is Jose Gonzalez and I'll be discussing the relationship between epigenetics and cancer development and making emphasis on the therapeutic implications of these studies. According to Tawfiq, in cancer, epigenetics have a crucial role into the growth and the progression of the tumor. So today we'll be exploring the modifications such as non-coding RNAs, histone alterations, and DNA methylations, which are key components of epigenetics. We'll be discussing how they contribute to cancer formation and their therapeutic implications. According to Costa, epigenetics refers to heritable changes in gene expression that occur without an alteration to DNA sequence itself. Key components are, like I mentioned, DNA methylation, histone modifications, and non-coding RNAs. Now, all these processes, they're involved in cellular processes and gene control and gene expression. Therefore, they have a great impact on disease and cancer development. Now, let's quickly define these components. DNA methylation refers to the addition of a methyl group to a DNA molecule. This is often associated with gene silencing or activation of the gene, depending on the context and the location of such molecule. Then we have histone modification, which involves chemical changes and alterations to histone proteins, which are proteins associated to DNA in the nucleus of the cells. They have a great importance in terms of structure and gene control. And last, we have non-coding RNAs. They are very important molecules that play a role in transcription, translation, and chromatin remodeling. DNA methylation plays an important role in preserving genomic stability. By preventing the activation of transposable elements and repetitive sequences within the genome, this is achieved. For example, hypermethylating these areas ensures the integrity of the genome, and this is how DNA methylation prevents cancer. Zhao and De Saulniers found studies where a correlation between appearance of cancer and abnormal patterns of methylation was found. For example, when they found abnormal DNA methylation patterns, the downregulation of tumor protector genes like p53 was observed, resulting in cancer development. This gained knowledge on DNA methylation in cancer is being used by researchers like Costa, who demonstrated that DNA methylation markers are being used to predict clinical outcomes and to see how well patients respond to treatment. This is huge when it comes to treating cancer. It's going to change the outlook on how we approach cancer therapies, whether it's be by a combination of these inhibitors or monotherapy. Having discussed DNA methylation, we move on to the next topic, which is histone modification. Histones are proteins that surround chromatin in the nucleus of cells. This histone can be modified by chemical alterations such as acetylation or methylation, and they're going to influence how accessible the chromatin can be to the transcription machinery. For example, the acetylation typically correlates to an open chromatin state that ensures transcription of the gene. On the other hand, methylation can both result in silencing or expression of the gene depending on cellular context. This is important to understand how they play dynamically and how they can be targeted to improve the results of cancer therapies, which is something that we're going to discuss here in this slide. For example, we can see the histones with the enzymes that are going to be targeted by drugs that researchers have created. These enzymes are acetyltransferases and deacetylases, HATs and HDACs for short. By targeting these enzymes, we ensure that we inhibit them, and then the acetylation occurs, and we have suppression of the gene, which prevents the genomic instability, therefore cancer development. Moving on, we also have a table that shows the main brand drugs approved by the FDA using the same knowledge that we have discussed so far. Li and Yan, they found that HDACs were very important in the realm of cancer therapy, not only because of their role in modulating regular processes, but they affect the immunogenicity of cancer cells themselves. For example, they found that high HDCA 137 and low HDCA HDAC 4511 mRNA levels were significantly associated to overall survival and disease-free survival in gliomas. Now we move on to the last topic, which is the non-coding RNAs and their role in cancer. Non-coding RNAs are RNA molecules that do not encode for proteins but do play an important role in gene regulation. They can be divided broadly in two categories, small and long non-coding RNAs. They both have, like I said, important roles in cancer development because they work at different levels, such as histone modification, transcriptional regulation, and post-transcriptional regulation. So they're going to be very important in the realm of cancer therapy and clinical outcomes, as we can see in the next slide. Here we have a study done by Barwell in 2020, where he studied the expression of GS5, which is gross arrest specific 5, a long non-coding RNA molecule, which plays an important role in apoptosis and other cellular processes. He found that when the molecule was downregulated, we had higher expressions of cervical cancer compared to when the GS5 was more expressed normally. The same phenomenon was observed when they compared metastatic to non-metastatic cancers, which you can see in the figure B. So we can move on to the next slide, which talks about the therapeutic implications. Basically, by detecting the cancer earlier, we can improve clinical outcomes, and this is where non-coding RNAs comes into play. According to Barwell, they have been found to be great biomarkers for cancers, so they are being targeted to and researched as to create a new outlook into clinical outcomes for patients. In conclusion, we saw epigenetic mechanisms such as DNA methylation, histone modification, and non-coding RNAs that have important roles in cancer development. We understanding these mechanisms allow us to create new strategies, whether it is to detect the cancer or to treat it, that has resulted in an improvement in clinical outcomes for patients. And this is only the beginning. I'm very excited for the future in this area. Thank you so much for the attention. You can reach out if you have any questions.